Hello, welcome to this video. So in this video we are going to talk about proteins. So what exactly are proteins? So proteins are polymers of amino acids. So this simply tells us to say amino acids are building blocks of proteins. So it goes like this. So you have amino acids. So you have amino acids. So when you join amino acids, that means you have a sequence. You are going to have a sequence of amino acids, which is, let's say, this is an amino acid, 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 this is another amino acid. So this is a sequence of amino acids. So this sequence of amino acids here, this one we are going to call it a polypeptide. So a polypeptide is joined by peptide bonds. Peptide bonds or amide. Amide bonds. So this is a polypeptide. So when you just have two amino acids like this, you have one amino acid and you join to another amino acid. So this bond here is the peptide bond. But since you have two amino acids joined together here, you're going to specifically call it a dipeptide, which means two amino acids in the chain. So proteins are actually categorized into three levels. So we have the first level, so levels of proteins. Let me just summarize it before we go into looking at the amino acids. So we have the first level. Level 1 we have primary. Structure. So we have primary structure. So the primary structure is just this sequence and the number of amino acids in a polypeptide. So we shorten it like this. So this is for polypeptide, short for polypeptide. Yeah. So this is just the sequence and then number of amino acids in a polypeptide. That's what we call the primary structure. So the bond that was the primary structure is just the peptide bonds. Then the other level we have the secondary structure. So the secondary structure involves coiling, of polypeptides to form helix, alpha helix, and beta pleated sheets. So these here, so for this one here, alpha helix, this one you just have intra hydrogen bonds. So just hydrogen bonds within one structure. But for beta pleated sheets here, you're going to have inter hydrogen bonds. So that means, okay, let's just try to draw this structure. So alpha helix, just like a spiral, it forms a spiral. So something like this. So let's assume these are hydrogen bonds. So we are going to call this alpha helix. Alpha helix. So going to have another one. Behave. So when you have hydrogen, we have hydrogen bonds, hydrogen bonds. So this is what we are going to call beta pleated sheets. So hydrogen bonds are involved here. Then we have the third level. So level 3, we are going to have the tertiary structure. So for the tertiary structure, this is just the folding of polypeptides into 3D structure. So we have different interactions here. So here we are going to have non-covalent interactions. And also there are disulfide bonds here. So the fourth level now, the last one. So the fourth level, we're going to have the quaternary structure. So the quaternary structure here, so this one involves polypeptides interacting with each other to form a functional protein. So for example here we have an example of a protein that we can find here. So we have like hemoglobin. Yeah, so let's go now. Since I was just giving a brief here. So let's now see the functions of proteins. Functions of proteins. So what exactly are the functions of proteins? So firstly, first we have provision of structure. Provision of structure. So e.g., you find the proteins in collagen. You have collagen of the skin, e.g. collagen. Then secondly, proteins are used for transport as transporters. E.g. hemoglobin. We have a protein that is hemoglobin, which transports oxygen in blood. So we have also for as enzymes. For catalysis. So catalyst, since enzymes are catalysts, so that means that these enzymes, they speed up the rate of chemical reactions, and they speed the rate of chemical reaction by reducing the activation energy. So the fourth one is for, they are used as hormones. As hormones, e.g. insulin. Insulin, e.g. insulin. So also for defense, for protection. Number 5, for protection or defense, e.g. antibodies. So the last function, I think these functions are enough. So the last function we are going to, for communication. So cell to cell communication is a function of proteins, e.g. cell receptors. So let's go now to amino acids. We start from scratch. Amino acids. Amino acids. So amino acids were already discussed in the introduction, to say they are building blocks of proteins. So we actually have 20 natural amino acids. There are 20 natural amino acids that are important. So out of the 20, there are those amino acids that cannot be synthesized by animals. Cannot synthesize some amino acids. And these amino acids are called essential amino acids. So this is for amino acid. Whenever you see AA, since I've already written in full here. So essential amino acids. But those amino acids that can be synthesized by animals, they are called non-essential amino acids. So most of the amino acids are actually alpha amino acids. They occur as alpha amino acids. So this just means that the, the amino group is attached to the, okay, let's write this amino group is attached to the alpha carbon. And the alpha carbon is that carbon which is attached to four different groups. So for example, we have glycine. Glycine doesn't have an alpha carbon. CC, the alpha carbon that is referred to glycine is attached to two hydrogen. So we don't have like four different groups that are being attached. So all the other 19 amino acids are alpha carbons or anomeric carbons. So let's proceed. So the general formula for amino acids we have, let's use a different color. Let's use a green. So we have the alpha carbon that we were talking about. So this carbon is attached to four different things. So we have the alpha, let's indicate it. So we have the R group here. So this R group, it can be any group which is the characteristic of any amino acids. So this group here, R group, is what differentiates the amino acids from one another. Like for glycine that I was talking about here, you're going to have the hydrogen atom here. So then here we have a hydrogen atom. Then we have a carboxyl group here. Then here, that's where now you're going to have the amine group. So we have H2. Yeah, so this is the general formula for amino acids, the general structure of amino acids. So this group here, this group, carboxyl group, is acidic. Then this one here is basic. Basic group. So when you have like two carboxyl group, so that means the amino acid is going to be an acidic amino acids. An acidic amino acids. So when you have two carboxyl group, let's even write it. So this signifies that a given amino acid is acidic. Since we've already noted to say the carboxyl group is acidic. So I already mentioned to say this R group here is what gives the characteristics of any amino acids. Let me just write it down. Is what gives unique properties for each amino acid. So most of the amino acids, so amino acids can exist as A, O, and D amino acids, but amino acids that are considered here that we are talking about here, all the amino acids like 20 amino acids exist as AO amino acids. As AO amino acids. And this means that the amino group is on the left side, as we can see from the structure that I drew here. It's on the left side. So this is the AO amino acids. So amino acids actually, when they are in the neutral environment, they exist as Zwitter ions. Let me use a green color. Exist as Zwitter ions at a neutral pH. Zwitter ions. So a Zwitter ion is a molecule that has a zero net charge. In this case, any amino acid that has got a zero net charge is called the Zwitter ion. So that means that you have a negative charge on the carboxyl Okay, let me just write it like this. So it will be like this. So we have a negative, then and the amino group. So we have H3. So that means you are going to have something like this, to be like this. So this means you have a zero net charge. So that's what we mean by Switter ions. Not only that, so amino acids, you find that they act as buffers. As buffers. And remember, a buffer solution is any solution that can resist greater changes to the pH. So this means that this means they can donate and accept protons, which are just hydrogen ions. So that's what we mean. So this means that the amino group, so the amino group can accept, as we saw here, is plus here. And the carboxyl group can donate H plus ions. So remember we said that the amino group, it's basic. So this means that it can accept protons. But the carboxyl group, we said it's acidic. So an acid is any substance that can donate protons. So that means this carboxyl group is able to donate the proton. So when it loses the proton, that means the charge goes down. Yeah, so I think we can end for this one here, so that in the next video we are just going to continue with the classification of amino acids, so that we know how amino acids exactly are classified and why we can call them acidic, basic, like neutral, just like that. So thank you for watching the video. Make sure you subscribe to the channel. This is Unza Dani.